So welcome all to this SIB virtual computational biology seminar series. Today we have the pleasure to host Daniel Wegmann from the Department of Biology at the University of Fribourg and also a member of the SIB. Daniel got his PhD in computational population genetics in 2009 with Laurent Escoffier, who is also a member of the SIB at the Institute of Ecology and Evolution at the University of Bern. From 2009 to 2011, he pursued his research with a postdoctoral training with John November at the Department of Ecology and Evolutionary Biology of the University of California in the US. Then Daniel came back to Europe and he did a second postdoc at the School of Life Science at EPFL here, next door in Jeff Jensen's lab. And then since 2013, he is an associate professor at the University of Fribourg, leading the Statistical and Computational Evolutionary Biology Group, which is also affiliated to the Swiss Institute of Bioinformatics. So the primary aim of Daniel's group here is to better understand the underlying evolutionary and ecological processes that have been shaping this diversity over the course of evolution on our planet. To achieve this, the group designs and evaluates new statistical and computational approaches to infer complex evolutionary histories. So for this, they develop and apply machine learning algorithms. They then apply these approaches to the wealth of data currently being generated, mostly in collaboration with experimental groups. And they are further committed to making all their development available to the scientific community by releasing easy-to-use software packages. I encourage you to have a look at the webpage of the Daniel's group on the website of the University of Fribourg if you're interested in knowing more about the packages. So today Daniel will take us, will tell us about the bioinformatics, how bioinformatics and population genetics can help tracing the spread of farming into Europe using ancient DNA. So Daniel, thanks again for accepting this invitation, and the floor is yours.
Thank you very much. Now we can have a mic transfer. Let's see how that plays out. I have to click here. Okay, this is the easy way to lose the people online, right? You just tap on a mic. Okay, thank you very much for the introduction and thanks for hosting me here. It's the first time I'm giving a seminar where, I don't know, half of the audience or so is actually virtually absent from the room. So I don't know, it's a kind of a weird feeling, but we're gonna go through. So I'm gonna talk about, as it was said, the spread of farming in Europe. But before we're gonna start, I just have like a picture here. So this is for the people listening online, in case you're wondering like who that person is that is talking to for the next 45 minutes or so. Well, it's me and this guy here that you see a picture of. And anyway, so before we're gonna head into all the bioinformatics and population genetics, I'm gonna just start my talk by actually giving you a very ignorant and super high-level summary of what I think was probably the most dramatic social or cultural revolution in the history of Europe, okay? And in order to better understand that kind of revolution, we're gonna just turn back the wheel of time about 12,000 years. And that was a time at which, when we were looking at Europe, most of the continent was actually colonized by societies that were basing their, or were trying to search for food by foraging. That's hunter-gatherers all over the continent. We by now know that many of these different groups are actually genetically quite distinct. There's a large diversity. Very little is known about that, but we just know that there was a large diversity, different groups, but they were pretty much all doing the same thing, that is running after animals and collecting vegetables or berries, fruits in the forest. Then about 11,000 years ago, something quite remarkable happened. That is, we see the first farmers arising in the Middle East, and more precisely in an area known as the Fertile Crescent. Okay. The change from foraging to farming is quite a dramatic change. It's not only the change in terms of what you eat, but with it comes a whole set of other things that shape culture. First of all, it's becoming sedentary. So farmers tend to live in one place. They're not mobile. They don't follow herds traveling around. They actually are in one place. And that is quite a change. If you think about it, it probably even changes the way we look at property and a lot of questions like that. On top of it, becoming a farmer means adopting a lot of technical things, a lot of new tools, and working with specific domesticated animals and plants. So definitely these cultures, a farming culture and a hunter-gathering culture, are very, very different. Okay? What we see then is that after farming kind of arose, it didn't took that much time to actually spread this way of culture throughout the continent. By around 8,500 years ago, the first farmers were appearing in like Western Europe or on the European continent, especially down here in the Aegean region, and establishing their first societies there. And then within a few other thousand years, all of the continent was pretty much covered by farming societies, and the last hunter-gatherers kind of disappeared around that time. So the question is therefore, how does this, or one of the questions we're interested in is, how did this cultural revolution actually play out? And there's really two different kind of theories you can put forward about how this could have happened. The first one is, if we just turn back the wheel of time a little bit again, so we have farmers kind of appearing in the Middle East, and then the first hypothesis just states that, well, the farmers, the hunter-gatherers themselves, they were kind of jealous about this awesome lifestyle and tried to copy the way of life and became farmers themselves. If that hypothesis is true, then modern Europeans are direct descendants of like the foragers or hunter-gatherers that lived in the same place, okay? And these people became farming spread to Europe as an idea, as a way of life, and not necessarily by people. The alternative hypothesis to this is that it was actually the farmers themselves that colonized most of Europe, displacing most of the hunter-gathering people, of course, apart from this tiny little area here up in Brittany, but apart from that, like pretty much replaced the hunter-gathering societies. If that hypothesis was true, then we as modern Europeans are direct descendants of the first farmers from the Fertile Crescent and not of the hunter-gathering societies that were living in this area before. Okay. Of course, you can imagine every kind of intermediate scenarios between these extremes. And one of the big questions, I think, from the last decades was trying to figure out how this actually played out. Looking at existing ancient DNA, we can get a glimpse of that this change in lifestyle was first most likely driven by the exchange or by the colonization through people. So what I show here is an admixture analysis. So this is analysis in which you try, for all the individuals included, try to assign or to estimate their proportion of their genome they inherited from a couple of groups. So here this analysis we run with two groups. So we try to partition individuals into two groups, and for each individual try to estimate how much of their genome they inherited from one of these groups. Okay, and this is all ancient DNA data that was existing before we started the study. In the lower part here, we have hunter-gatherer individuals, followed by early Neolithic people, so the first farmers that actually colonized Europe, followed by samples taken much later in time, in the middle and late Neolithic. What we can see is that genetically, it's super easy to actually distinguish hunter-gatherers from the early farmers. They are really completely different groups. That's suggesting, therefore, that it was really people moving into Europe, bringing agriculture with them. And it was not the hunter-gatherers that were actually picking up the lifestyle of being a farmer. However, very interestingly, in later periods, there's kind of a resurgence of hunter-gatherer ancestry, suggesting that before hunter-gatherers disappeared, there was quite some gene flow between the two groups, so that in modern-day Europeans actually have some of their ancestors from both groups. Okay? However, even if we know kind of that now the spread of people was really, really important, it is still pretty much unclear where exactly these farmers came from. We know that farming was developed in the Near East, but the question was, were these early farmers that we see in Europe, were they the descendants of actually these people in the Near East or not? One major reason for why we do not know this, or we didn't know this, is that actually the preservation of DNA in ancient bones is a lot affected by climate. The cooler the climate, the better it's preserved. And it's just a very, very difficult task to actually obtain ancient DNA from warmer regions. And so when we started out this study, there was actually the southernmost kind of sample in Europe were all from the Balkans, but there weren't any samples available further south. Oops. Okay, for some reason, I'm backing a slide here. So what we then did is we started out by actually analyzing samples from the Aegean region, as well as from Anatolia, and trying to see whether these samples were actually in some way related to the modern samples that we have in Europe, okay? And this is work that was mostly led by Joachim Burger and his group in Mainz. They have like one of these super sterile, super clean rooms to work with ancient DNA. The issue is that if you actually touch an ancient bone, there's probably more of your DNA on that bone, that original DNA in the bone. So it's a really challenging task. And so these guys were working really hard to get libraries out of that, okay? This is then the data that we got in our lab, and we were trying to analyze to answer some of these questions. Before I'm gonna really delve into how we actually did some of these analyses, I wanna just walk you through some of the major characteristics of ancient DNA, okay? And these characteristics, they really influence the way we can actually deal with this data from a bioinformatics point of view. The first thing is that if you work with ancient DNA, most of the data sets, they have really, really low coverage or sequencing depth. There's multiple reasons for this. The first one is that we generally get extremely low DNA content out of the bones, okay? So what I showed you here on the right is for each of the samples, or I should say for the 5 best samples that we analyzed, so the people that screened up to almost 100 samples, and then identify the 5 most promising for sequencing. And so these are these 5 most promising sequences. And what you see is on the x-axis is the amount of endogenous DNA. That's the amount of DNA you retrieve that is actually human, most likely of the person that we want to analyze. All the other DNA comes from soil bacteria and all other things you sequence, but it's actually not human, okay? So in the case of, for instance, this Rev5 sample, more than 80% of what you sequence is not human. So that means if that was the only problem, just to get the same amount of sequencing depth as for a modern person, you would just have to sequence 5 times more, right? But there's more challenges associated with this. The issue is that because DNA content is very low, if you actually generate a sequencing library, you get a lot of the times the same fragment. You have to amplify the DNA using PCR, and you're gonna sequence the same fragment multiple times. What is shown here on the y-axis for each of these samples is the amount of unique fragments we're actually getting. Down here is a bunch of blanks that were run in the lab. So if you prepare a library, or if they prepare a library in this ultra-sterile clean room, they sequence here about 10 to the 5, so about 100,000 fragments, unique fragments, anywhere in those libraries. It's very hard to be so clean to have no DNA at all in your library. If you compare that to the worst sample in this respect we have, so this is bar 8, it's about 10 to the 7, it's only about 2 orders of magnitude more human DNA in a kind of library prepared with an actual sample compared to one that is just prepared from what we think was pure water at the start, right? So we get so little DNA out that it's really hard to actually have a lot of different fragments. And as a result, again, you have to invest quite a lot of money, quite a lot of sequencing lanes to actually push up coverage, okay? The next issue we're having is that because the DNA was lying in the ground for a very long time, it's usually extremely fragmented. So this here shows the distribution of the lengths of fragments that we got for 3 of our samples. And these were samples sequenced, 2 of them were samples with 2 times 150 base pairs paired-end library, right? And so we can estimate the fragment lengths exactly. And what you see is that even though we could have fragments up to 300 base pairs, the vast majority of fragments was around 40 base pairs in length. So the DNA is already extremely fragmented in the ground. And so even if you invest kind of a lot in having 2 times 150 base pairs or something, you're anyway just getting something around 40 base pairs per read, okay? As a result of that, the later samples were just sequenced with single-end kind of 100 base pair reads because it wouldn't make much of a difference, okay? So coming back, not only are you gonna sequence the majority of reads you're gonna sequence, not from the sample you're interested in, those that are from the samples are probably half or even less in length than the other reads, like further kind of lowering the amount of interesting DNA you're gonna get out by this approach, okay? Now the last kind of issue that is common when dealing with ancient DNA is the presence of so-called postmortem damage. So postmortem damage, these are chemical modifications that occur along the time to the DNA molecules that result in certain changes, okay, or mutations that are not really reflective of the person's DNA or the person's kind of diversity that we're interested in, but are just areas that are kind of compiled over the course of time. The most common form of postmortem damage is through the deamination of cytosins, okay? And if you have a deamination of cytosins, that turns into uracil, and if you do a PCR, that turns into thymine, okay? As a result, you're gonna see a lot of T's instead of C's when you look at these ancient fragments. And clearly, if we were to believe that these T's are all real, we have a lot of mistake in our genotyping, and as a result, may actually completely get the wrong type of impression of relationships among people or even the diversity. it's interesting to note, important to note that actually all these C to T changes, they appear also as G to A changes on the opposite strand if you map. So these are the two types of errors we see. Luckily, these errors do not just occur randomly, or I mean, they do occur randomly in the fragments, but what we see is that there's still a certain type of distribution that comes with the position in the read, okay? So what I showed here in the picture are these type of distributions that we inferred for two of our samples. The way I inferred these numbers is by simply comparing, by mapping all the reads against the human reference, and then just counting how frequently do we see a T in one of these reads if the human reference was a C. Now, certainly, you do expect a real difference between a human that you look at and the reference. You do not expect a person to always be exactly like the reference. So there's a certain fraction of sites that you do expect a true difference in which the individual would have a T, but the reference has a C, okay? That's, I don't know, something like 1 in 1,000 or 1 in 4,000 base pairs, you would actually expect that, okay? However, what we see here is that at the beginning of a read, so on the x-axis we have the position, at the beginning of the read, in certain cases, more than 50% of the times in which a map maps against the C in the reference, we actually see a T. And that's exactly this post-mortem damage that I was mentioning before. And the reason for why this kind of really accumulates here at the beginning of the read is because this chemical modification happens much more likely, or is occurring much more likely if the DNA is single-stranded. So we have the DNA fragmenting up into fragments in the ground, losing some nucleotides at the end. We get sticky ends with single-stranded DNA, and that's where these chemical modifications appear. This is known because we can actually replicate that process in the lab. You can just expose DNA to a lot of kind of weird influences, and you can replicate that process. So the chemical way by which this happens is quite well understood. Okay, and you see that this is a common pattern in all of kind of the libraries we looked at. There are different types of libraries you can use for ancient DNA. We can have paired libraries here in black for Ref5. You can treat them with a chemical that actually turns uracil back into cytosine before sequencing. So you would do that, apply that chemical first before doing PCR. And like that, you would actually lower the amount of these kind of changes. They're still somewhat present, but to a much lower degree. The downside is you actually lose a lot of material. So that's why we stopped kind of doing this. Then for bar 8, we use single-end sequencing. There it's just interesting to see that because the reads do not actually necessarily reach the end of the fragment, they are at the beginning affected by postmortem damage, but all the reads that actually do not reach the end of the fragment, they're not affected on the other side. That's why you see kind of here in the lower panel in blue that those reads are actually exactly as long as the cycles we used in the Illumina machine. So if you sequence 100 base pairs, your read is exactly 100 base pairs long. Then that means that most likely these reads did not reach the end of the fragment. And if you look at those, we see a reduced rate. But other than that, this is pervasive in the data we analyze. Okay? So these are three things. We have very low amount of data generally, and we have these errors. Okay? And now we are interested in working with this data to understand or to infer genetic diversity in these ancient samples and compare them to modern samples. Generally, low sequencing depth just means ambiguous genotyping. Okay, that's something we're all aware of. That's why people try to increase depth. Okay, but what it means for us is that we cannot simply increase depth. We have samples which have less than 1x sequencing depth, so coverage, less than 1x. And they were already sequenced on many, many different lanes of Illumina. So money is not really going to solve this problem in the short term, right? If you just want to say, oh, let's go up to 10x, it becomes unaffordable even for super rich labs. So we have to deal with the fact that some of these samples have depths of around 1x. Okay. Clearly, if we are interested in inferring genetic diversity of a sample that has just a 1x coverage, then by first calling genotype and then just counting all the sites that are heterozygous seems to be a pretty stupid idea, right? So we have to do this in a somewhat different way. In population genetics in the recent years, there's been a lot of tools proposed for actually trying to try to infer population genetic parameters or statistics without calling genotypes. This is this whole idea that we can invest money rather in more biological samples rather than just in the technical replicates we do for a single sample, because all these different technical replicates. One of these commonly used tools is ANGST, stands for Analysis of Next Generation Sequencing Data. And this offers an algorithm that actually infers allele frequency distributions or site frequency distributions, SFS, without ever calling genotypes. Okay, and they show that you can actually infer these distributions even if you have coverage of 1 to 2x, usually for a sample. However, ANGST is not really applicable to our data out of two reasons. First, it requires a priori knowledge on which are the major and minor alleles in the population. Now, clearly, we have a lot of knowledge about, say, human populations from Europe. We could actually know which ones are most likely going to be the major and minor alleles. But we're very careful in the sense that we don't want to bias the genotypes of our ancient samples through modern knowledge, right? Otherwise, they're just going to appear more modern than they probably are. So we want to have a way to infer things that does not require knowledge from modern populations. The second problem with ANGS is it does not actually handle postmortem damage. And if we don't handle that, we're going to grossly overestimate diversity, okay? So we needed kind of a different approach. So this is when we came in, and together with a postdoc in the group, Athanasios Kousatanas, and a PhD, Vivian Link, we developed methods to actually analyze this data inferring genetic diversity. Okay. Now, since this is a bioinformatics series, I'm quite excited because I can actually show some equations, and I'm sure that at least a fraction of the audience will really appreciate that. That's not the case if you give usually talks to different audiences, so I'm gonna benefit from that by actually having a couple of slides with a few equations, okay? So bear with me. Because at least I'm excited about it, okay? So the model we propose to infer genetic diversity is relatively simple, okay? We're just interested in the genetic diversity of a single individual. You can call this heterozygosity. A population genetic statistics for that would be zeta, okay, which is given by 2t mu here. So what is this? 2t mu, so 2t, t is the time to the most recent common ancestor of the two sequences of a single individual. So if these are the two sequences in one individual, these two sequences do have a common ancestor, let's assume t generations ago. The total length of the tree connecting the two is 2 times this time. And if you apply by the mutation rate mu, we get the amount of mutations accumulating on that branch, okay? So that's the zeta we're interested in estimating. We will never be able to estimate either a mutation rate or t separately, okay? However, like it clearly puts a model in place in which we can explain like the genetic diversity of a single individual that we're going to use here. In addition, we're going to say, okay, we don't want to use any modern information, but we're going to use the sequence composition in the region kind of as information about what type of alleles to expect. If you're in a GC-rich region, you're more likely to see G and C alleles than A and T alleles. So neighboring loci do tell you something about the probability of seeing a particular allele. So that's why we're going to say we have regional allele frequencies pi here. And these frequencies, they obviously sum to 1, but they give us some information about what type of alleles and mutations to expect in that particular region for which we want to infer genetic diversity. If we have these two, we can build an easy model. Okay, here just put down the likelihood for this. So likelihood in which we have a product across each site, assuming that mutations are independent or between sites. That's an assumption generally made. I think it also makes sense. Just because my granddad had a mutation at one locus does not prevent my grandmother from having a mutation at a locus just nearby, right? So we can assume that mutations are independent between sites. So we can take the product across sites. But then for each site, because we do not know the genotype, we're going to just integrate out the genotype. We're going to integrate over the genotype and just ask what's the probability of the sequencing data, di, given the genotype that I integrate over, times the probability of the genotype given the zeta and pi that I have. Okay, so this way, the genotype is a latent variable they're going to integrate out, and I'm interested in finding estimates of both zeta and pi without having to know anything about the genotype in between. That's the basic idea. So in the next few slides, I'm just going to walk you through how we actually, through the model that we really employ for these two terms. So first, what we call the emission probability. That's the probability of the sequencing data given the underlying genotype, and then the probability for the genotype given the parameters we want to know, okay? So for the substitution model, that is the probability of the genotype given pi and zeta, we just employ the classic Felsenstein substitution model. It's a famous Felsenstein 1981 model, very, very commonly used in phylogenetics. It has some advantages over other models, namely that it actually allows for recurrent mutations at the same site. Okay, the model is, in that regard, though, very simple. The formula may look a little bit complicated, but if I walk you through, you see it's actually a very simple model. So what we have here is the probability of a genotype given the frequencies pi and the heterozygosity zeta. Zeta is, we can see, as a kind of an overall mutation rate in that sequence. So if you look at the lower line here, if the two nucleotides of a genotype are actually different, so k not equal to L, then in that case, we do require at least one mutation on the sequence, okay? And then what we have here is the probability that there's at least one mutation is given by 1 minus e at the power of minus the rate. Okay? And then if that's the case, we just multiply with the probability that if you follow a tree, the first nucleotide on one side is actually of type K, and the last nucleotide on the other side is of type L. So it gives you pi K times pi L times the probability of at least one mutation. If the two nucleotides are equal, that is it's a monomorphic site, then there are two options. One option is that there is actually no mutation, okay? So that would be e at the power of minus zeta. Or that there is at least one mutation, but that the last nucleotide to which you mutate to is the same as the first one, okay? So this is why we multiply this again with zeta k. So it's a very simple model, allows for recurrent mutation and has just the parameter zeta plus pi that we require for our model. The emission probabilities are best explained with a little example. So consider a case in which one read at one position here has the data dj, so d is the data and j is just one position in the read. So we see here an A nucleotide being read. And we ask what's the probability that the read actually turns out to be an A if, and now here I'm just gonna, for simplicity, assume we have a haplotype case, so we have a genotype that is just a T. Now we're just gonna assume that in this haplotype case, then this means that there is a sequencing error happening at this location. And a sequencing error happens with probability epsilon j. And because a sequencing error can turn a T into an A, a C, or a G, it's just in 1/3 of the cases, and that actually turns out to be an A. Clearly, this is just for the haploid case. You can spin this further for a diploid case. It's not very hard. I just use this as an example to see where actually postmortem damage now comes in. Because if we have the same, again, haplotypic case in which the underlying genotype is a C, but the data that we read is a T, then there is now like more than one way by which you can actually end up having a T in your sequence if the underlying genotype was a C. One of the ways is easy. It's again like a genotyping error, a sequencing error. So that's the second term here. It's epsilon third times the probability that there is no postmortem damage. So that actually the DNA that I get from the bone still contains a C, and then the T appears by the machine making a mistake. Or I have the probability that I do have postmortem damage D, so that the DNA molecule was actually the C was actually turned into a T before I even machine is not doing any mistake and everything is fine. So I'm having 1 minus the probability of a sequencing error. You can spin it further. You can develop this for all kind of genotypes that you want. We did this for all the diploid genotypes that exist, okay? And then if we have this, we can actually build a model by which we take this, or we take these emission probabilities directly into account when integrating over genotypes, okay? The question is now from where do we get the probabilities of the postmortem damage, so DJG here? Well, for this, we actually can use the exact same, the curves that I showed you before, where we see a pattern of this exponential decay and actually just fit the generalized exponential decay function to it, and then we can assume that this is the rate we have. So a priori, we estimate this from the data and then assume this to be known, okay? Once we have figured out how to do these probabilities for, we can easily assume that the individual reads at one location are independent, so we just have to integrate over these different reads. We just have to take the product across all the individual data points that we get for the same site to get our likelihood. So how does this actually perform? Like, does this work? Can we estimate zeta? What type of sequencing depths do we require? So these are some results we got from simulations. We first simulated data in a 1 megabase window for different coverage or sequencing depths here. So from 0.2 to 3.2. And what you can see is that as soon as coverage is a little over 1, we seem to get quite accurate estimates if zeta is at least 10 to the minus 3, which is what is the average in the human genome, roughly, okay? So we seem to get quite accurate estimates, even if coverage is only slightly above 1x, if we combine data from about 1 megabase. Similarly, we can simulate 1x coverage, but then ask how does the window size affect things? So how many sites do I require to accurately infer heterozygosity if the coverage has a certain number? I should say, like, this is not coverage per site, but it's the average coverage we simulate in the window. Again, we see that if the window size approaches about 1 megabase, we get quite accurate inference. If we have much less data than that, there is some bias that we see, okay? So generally, 1x coverage, about 1 megabase, 1 million sites are required to do that. Interestingly, the window size as well as the sequencing depths, there are two factors that seem to work jointly here together. You can either have larger windows and less coverage, or less coverage and larger windows. One way to plot this is here in the last plot on the right, where I plot for all combinations of window size and coverage the amount of error we make in the estimation. And you see this is from simulation, so it's kind of like a wiggly density plot, but what you should see is that for all combinations of window size and coverage that result in about the same number of sites, the same number of individual read information being there, you get the same performance. That means that whenever we are interested in inferring genetic diversity from an exceptionally low coverage sample, we can just increase the window size. We may not be able to infer heterozygosity on a 1 megabase window scale, but just 10 megabase window scales, and then it should just be fine. If we have a sample with a lot more data, then we might actually just zoom in and get those estimates at a much finer scale. Clearly, the performance of this is affected by the actual diversity in the sample. The higher the diversity, so the higher the zeta here, the less data is required for proper inference. I think that makes kind of sense. If in a 1 megabase window you have a single heterozygous site, you need quite a lot of data to be confidently actually seeing that. However, if every second site is heterozygous, you don't need that much data to realize that there's plenty of diversity in that window. So we see this relationship, and that means that for very low zeta values, like 10 to the minus 4, we need slightly higher, maybe coverage of 3 if we were using 1 megabase windows, or actually we might just use higher windows if we estimate zeta to be relatively, or we expect zeta to be relatively small. So that seems to work quite fine. We were quite excited and then just took our 3 Greek samples here. So these samples are up to about 10,000 years old. We had sequencing depths between 0.8 for the worst and 3.5x for the best sample. And then we kind of run our estimator. So these are the results we got, and compare them to our expectations. So the average human genetic diversity is about 10 to the minus 3, as I said before. Now, do you see, it's like we get super different estimates. Like we get this blue guy up here, which has something like 10 to the minus 2 or even more. That means 1 in 100 base pairs seems to be heterozygous. Well, this red guy is like this kind of jumping up and down between 2 values. One is 1 in 100,000 and 1 in, I don't know, something like 10 billion sites being heterozygous. Now, that should appear to be a little bit strange, right? I mean, in the end, these are all humans. It's just 10,000 years ago. Even if you go nowadays and go to the most distinct human populations that you're going to sample and look at, it's hard to find anything like that in diversity. So there's a real question, like, why are these estimates so different? We don't expect them to be so different. In the end, they even come from, we think, from the same population or same region at the same time, right? About pretty much the first farmers arriving in the Aegean. Why should the first farmers differ so much in their genetic diversity? Okay, so the answer is actually that the answer is that these are not very good estimates, but there's something else in the data that prevents us from getting proper estimates. And what it is, what we figured out quite quickly is that all these kind of all these tools that try to estimate diversity without calling genotype, they require an accurate information about sequencing errors at a particular site. We do take quality scores into account, right? And if, of course, these quality scores are completely off, we're going to get completely different diversity, completely wrong diversity. So the point here is that I want to make is we do need to recalibrate quality scores urgently or absolutely if you want to estimate genetic diversity. If the sequencing machine tells you, I'm very, very sure that this is the base you're going to see, but it's actually wrong, you're going to have very biased estimates in terms of diversity. And as you will see, we can explain these differences solely by the issue with recalibration. The question is just, how do you recalibrate data if you have this much data? How do you really recalibrate that? And if you don't want to use modern information to do that. Most current recalibration tools, they require information about which sites are actually polymorphic in the genome, which one is the reference site, and so forth. And we don't want to actually make our ancient samples more modern just by imposing all that information. So we were therefore thinking hard about how could we devise a recalibration strategy that does not require any sort of reference information. Okay, now here's our solution. We're relatively excited about that because it also works, of course, for all the species for which you do not have that information available anyways, right? If you're working in a non-model organism, you're the first one to sequence it. There is no kind of dbSNP around. There's no perfect reference genome. So how are you going to recalibrate with GATK or BQSR tools, right? You don't know. Well, I think this is one way maybe by which it's feasible. So I'm going to walk you through. The thing is that we make a particular assumption, namely that we can get one part of the genome for which we may not know the sequence, but we do know that it should be monomorphic all over. Okay, and one of these type of sequences is, for instance, X-linked data in males. So it happens that most of our Greek samples were actually males for pure chance, because in the screening it was pretty much 50-50 regarding the sex, but for some reason we got like mostly males in this sample. And so if we just look at the X-linked data, then all these sites, they should be, we don't know what their allele was on the X, but we know that they were monomorphic. One copy of the X, so we should never have a heterozygous site. Now we can use this information in the following way. So we're going to assume that there is some sort of transformation that transforms the machine quality score that we get into the true error rate. Okay? And this transformation, we put down here these lower equations in two ways. So first we're going to say that there is this a new here ik that is going to be a linear combination, okay, of what we call q, which is some sort of information that we can turn into what we call this nu, which is then transformed with a logistic function into an actual error rate. So the logistic function is just there to bound the error rate between 0 and 1. You cannot have an error rate below 0 or above 1, right? So this is why we use this. And the linear function here is just a combination of information that we get for every base, such as, for instance, the actual quality tells me it is, but also the position in the read, whether the position just before, what type of nucleotide that was, and all these kind of information that also BQSR used. But we use this here in a linear function, okay? Once we have that, we can build a likelihood function for our haploid data. So it's the first equation here, where we express the probability of the data given these coefficients beta that we want to estimate of our linear model, okay? Here in this way. So for each site, we're going to first integrate over the unknown hidden genotype, because we do not know the genotype. But then if I pretend, say, let's say the genotype is A, then I'm going to go through all reads that cover that site. I'm going to say, what's the probability of seeing this data here, given that the genotype is A, the error rates that I have for all my base pairs, okay, and all the PMD probabilities that I have my base pairs. And then the product of this multiplied by the probability that the genotype is actually A, right? And then the sum over all possible genotypes. We're just going to integrate out all the genotypes. And then I want to know which are the coefficients beta of the linear model that maximize this likelihood. Okay? So I try to find the transformation of quality scores that best explains the data in a location at which I do not expect to see any polymorphic sites, but without actually making any assumption about what the reference base is. Okay? So this is the basic idea. And so we use an EM algorithm to work that out. It works relatively fast. Does it work? So this is, again, from simulated data. So here we just simulated data where we distorted the qualities using 4 coefficients. The first one is the actual coefficient distorting using the actual quality score of the machine. One where we take the square of that. That's just if you have nonlinear effects, we can put that in with a square. And the position, as well as the position square. Okay? In practice, we also add all 10 different contexts that you can imagine as additional factors. So this is then we then run our tools or our toolbox to actually estimate these coefficients from simulated data, again, within a 1 megabase window for different sequencing depths. And it seems we need a little bit more data than for CETA, but if you have something like 2x coverage in a 1 megabase window, it seems to be possible to estimate these coefficients quite nicely. Or if you have lower depths at 1x, so this is data not shown, but if you have only 1x, you need about 10 megabase data, then you're on the safe side, and you can actually estimate these coefficients very, very reliably, okay? So we applied this to our Greek samples. This is what we're gonna get. So this is the quality transformation table, where on the x-axis we have the quality score reported by the machine. On the y-axis, the one that we get after our recalibration, and the colors here represent density. And what you see is that for all these 3 samples, we actually find that the qualities given by the machine are too optimistic, right? The machine always tells you, for instance, here, quality score is 40, but then this would be the line here, the dashed line is when both qualities are the same, since all these points are actually below. So all the high quality sites, in particular, are actually not of that as high quality as the machine suggests, okay? So we needed to recalibrate that. You also see that the pattern is quite complex. It's not like kind of just a weird line. That's also because there's lots of different libraries sequenced on many different lanes and even different machines. And of course, for each library and each lane, we had to recalibrate on their own, okay? So you cannot just recalibrate putting all the data together, but we recalibrated for each library and on each run independently. That's why you get these complicated pictures. But anyway, we see a difference in these, in the quality scores. And if we then use these recalibrated quality scores and now infer genetic diversity for our samples, things suddenly start to make a lot of sense, okay? So here is just an example. We, of course, downloaded all the other type of available data, ancient data as well, and started to play with this, especially all the males, because we know this works for males. Okay, and so I should say, like, we have some strategies for females now. We use ultra-conserved elements. We also tried mtDNA. It's just pieces where we believe there's no polymorphism, right? And that seems to work. So far, we only had mostly males, so we're quite happy with this. We still have to experiment a little bit how to do this with females, but we're quite confident that ultra-conserved elements should actually be sufficient data to run the same strategy there too. So here, I show you in the first panel a trace of heterozygosity or diversity along the genomes. It's just a piece of chromosome 1. And so along the genome for different samples, we have TSI2 and GBR2. So these are just 2 male samples from the 1000 Genomes Project. And in comparison, we have KK1 and Bichon. So these are 2 hunter-gatherer samples. Bichon is from a Swiss cave, and this one here, KK1, is from the Caucasus region. Okay. So what we see is the trace here along the genome. Interestingly, the 2 modern samples, they almost fall on top of each other, right? It's not only true that if you take a British sample and a TSI from Tuscany, a Tuscan sample, they do not only have roughly the same genome-wide average in their heterozygosity, shown here as a dashed line, but even if you just follow kind of 1 megabase by 1 megabase through the genome, they're almost identical, these 2 samples in their diversity. However, interestingly, the hunter-gatherer samples here, they're a little bit different. They have genome-wide slightly lower heterozygosity, which you would expect given the lower population size, and their traces look a little bit different too. We then run this on a bunch of samples. So we have here 3 modern samples from Italy again, from Britain. We have BR2 is a Bronze Age sample. We have the 2 hunter-gatherers. Then we have 3 African modern samples and an ancient modern sample here, the Motta sample. What we find is like that this is shown the distribution of heterozygosity in the windows. We see that the Bronze Age sample seems to have pretty much the same diversity as a modern European. That's kind of expected. Hunter-gatherers have slightly lower diversity. We see that Africans have a higher diversity than Europeans. That's well characterized. Interestingly, this ancient African also exceeds modern Europeans in diversity as well. Right? We can then ask how correlated are the distributions of diversity in the genome. So if we just go along the genome and have the trace that I show here in the first picture, and we then do a correlation among that. So here is a Spearman correlation, and then try to get the pairwise correlations shown here as a tree. What we see is that we have all the modern African samples, the modern European samples clustered together quite nicely. Basal to this, we have the Motta sample here to the African. We have the Bronze Age sample basal to the modern Europeans, suggesting that they are not only very similar to modern samples in their overall diversity, but also in their distribution of diversity in the genome. However, the 2 hunter-gatherer samples, they're really basal to this, so they have a very, very different pattern of diversity in their genome. And interestingly, even between them, the difference is about as big as between any of them with any of the other samples. Suggesting again that these 2 hunter-gatherers actually come from completely different groups, right? It's not just that they are 2 old samples, they're representing completely different groups. It's maybe not as surprising, one from Switzerland, one from the Caucasus region. It's quite far apart, right? But it's kind of interesting that we pulled this up. Okay, so this was kind of our confirmation. Things seem to work quite nicely. Of course, we can then use the very same kind of model that we developed for this heterozygosity calling also for genotype calling. So we also implemented a genotype caller for ancient DNA that takes post-mortem damage and everything into account. If we do that using our MLE callers, we see that we can easily outperform GATK, even if GATK is actually using map damage tool, which is another tool to cut off PMD affected sites, okay, as we did here in the simulations. See, this is the amount of errors that we make here as a solid line. Okay, for Atlas, this is our tool. It's analysis tools for low coverage and ancient samples, how we call it. And then this is for GATK. We see, interestingly, our caller does not seem to have a reference bias. So if you compare our line here for reference homozygous calls or alternative, we see exactly the same pattern, while GATK clearly has a reference bias. So you're making much less mistakes if the true genotype is reference than if the true genotype is alternative. So the only way by which GATK is beating us here on the reference is actually by calling too many things reference, okay? You also see this in the amount of sites where there is no call being made by the tool. So GATK has a kind of large number of sites at which it just does not produce a call. And while we kind of accept, or we are willing to call a lot of sites, even if that may lead to a slightly higher error rate in the reference case, but our error rate is lower in the other cases, even for low coverage, okay? So clearly for ancient samples, it's easy to outperform GATK, and for those of you using GATK, just be aware of the fact that we're not the first ones to notice that, but it has quite a strong reference bias, actually. So most of your errors are towards the reference. Okay. So now that we have genotype calls and we have diversity and everything, let's go back to the biological question we started with, right? Now, if we apply all these tools to our ancient data, what can we learn about how farming actually now spread from the Fertile Crescent to Europe? Okay, so yeah, this is the slide that should have been at the very beginning. It's because I stitched together LaTeX and other slides. It's a bad habit. But anyway, let's jump to the PCA here. So this is the PCA we did on our cold genotypes at the SNPs, for which we have a lot of modern samples available as well. So this is a reduced number of sites. This is a PCA run on all modern samples. So we have different groups. You see quite nicely that if most especially for Europe, but most regions, if you do a PCA on genotypes of modern samples, you pretty much get kind of a map, because isolation by distance is the predominant factor explaining differences between humans, okay? We see here we have the Sardinians down here, Basque, Southern Europe, Central Europe, the Brits, Slavic people, okay? They kind of line up all around here. South, down here we have the Mediterranean, and then it goes towards the east. Now the question is, where do our ancient samples, we can project the ancient samples on this. We have much less SNPs for the ancient samples, and all ancient samples have different sets of SNPs. So you need a way to actually project them on the map. It's called Bruscostis analysis. And if we do that, okay, these are all the samples that existed before our study. So we have the European foragers showing up here. We have the early farmers down here. Again, confirming that these two groups were really distinct groups, right? But that we know already. The question is now, where do our Aegean farmers fall? And they actually fall right into the group of early European farmers. So we can say that genetically, the very first farmers that showed up in Greece from the Middle East, they are genetically the direct ancestors of the very first farmers in the rest of Europe. So we can trace back the genetic ancestry of the farmers, at least now, to the Aegean region. Okay? We see that there were hunter-gatherers living in Europe, people moving in. Okay? These people that we see establishing new farming settlements in Central Europe or Western Europe, these are genetically identical to the very early farmers we see in the Aegean region. Okay? But now, how can we trace that even further back to the Fertile Crescent? The answer is no, because our first Iranian farmer that we have, there's one sample from Iran, shows up here. So it's in a completely different place, suggesting that our Iranian first farmers are genetically not related to the Aegean first farmers. Okay? We can further look at that using a different type of analysis. So this is analysis done by Garrett Hellenthal, a collaborator of us on our calls. And so he's running a tool called Chroma Painter, in which what you try to do, so these are our two ancient samples. This is bar 8, our highest quality sample from the Aegean, and WC1, our highest quality sample from Iran. And for both of these samples, you try to explain the haplotype structure you see in that sample as a model in which you copy haplotypes from modern populations. Okay, so you say this is bar 8 here. So you say along the genome, you copy together haplotype, the best matching haplotypes in all locations from modern populations. Okay, it's kind of one of these admixture analysis. And then all these round dots are modern populations, and their size and color represents how much of haplotypes are called from that population. Okay, and you run this for all population at once, and then for bar 8, and then everything for WC1. And then what we see is, like here, the relative contribution of the populations to these samples. Okay? What we find is that our Aegean sample here seems to be, in terms of haplotypes, not only individual sites, but haplotypes, extremely similar to modern Europeans. Because you can explain the chromosome best by stitching together haplotypes seen all over Europe. Okay? On the other side, our Iranian sample is best explained by stitching together haplotypes we find in the Far East. Okay? So that is mostly in India and Pakistan and modern Iranian populations. So what does this suggest? This suggests that the Iranian farmers are actually not the direct ancestors of our Aegean and therefore European farmers. They may be the direct ancestors of the farmers that spread towards the east. But these two groups, the Aegean farmers and the Fertile Crescent farmers, they are genetically distinct. They're different. So what does that mean? We're not really sure. Does that mean that there were just multiple groups developing farming altogether? Does this mean that these farmers here learned or kind of picked up farming from these other people? We're not really sure. We think the latter one sounds quite plausible. So that's why I think we can conclude that the spread of farming from the Fertile Crescent to Europe involved two different kind of factors. First, farming most likely spread in some way by actually a cultural diffusion, be it because in that hunter-gatherers learned to farm from other farmers, or be it that there was a bunch of different groups developing farming together, exchanging ideas. We don't know. And then once these people that started to farm reached European shores, though, they were just spreading endemically and colonizing the rest of Europe as people, okay? So as a result, ancient DNA tells us that the spread of farming into Europe is complex, and it depends on which region we look at. From the Aegean to Europe, it's mostly by the replacement of people, but from the Fertile Crescent to Europe is a much more complicated story that mostly involved, at least to some degree, cultural diffusion as well. Okay, so let me sum up some conclusions. First, some general conclusions that I think we can learn from working with this type of data and apply it maybe to other data. So first of all, errors are an unavoidable part of NGS data. All of you working with NGS data, you know that, and we have to find a way to deal with it. Just ignoring it seems not to be a very clever strategy, as we've seen at least in this case. Filtering introduces a lot of biases, and if you're interested in genetic diversity, it usually introduces a bias towards too little diversity. If we're only willing to accept heterozygous sites or alternative sites where we're really sure, it means we have too few of them and we can underestimate our diversity. The trick that what most people in population genetics are kind of moving to is that we try to avoid calling genotypes altogether and just rather use tools that estimate what we're interested in by actually integrating over the genotype uncertainty. If we do that, we have the benefit of actually being able to work with low coverage data. And if we work with low coverage data, it means we can invest in more biological samples rather than more reads of the same thing of one individual. So investing in biological replicates rather than technical replicates, as I said before. However, whenever we do this kind of hocus pocus, we require that the quality scores we're working with are actually reliable. We can only integrate over genotype uncertainty if our uncertainty of the genotypes are properly estimated themselves, okay? And we have learned the hard way here that recalibration is super important if we work with low coverage data, to the degree to which our diversity estimates were pretty much useless without a recalibration before. Now, applying all this kind of logic to the ancient DNA case here, so I presented you some tools that we implemented in Atlas, this analysis tools for low coverage and ancient samples that we developed. It's a tool to specifically designed to work with ancient DNA particularly. Ancient DNA has two major things to deal with: low sequencing depth and postmortem damage, and both have to be properly taken into account. I showed you how we developed a reference-free base quality score recalibration. I think that might also be interesting for people working with non-model organisms, for instance. That we can have we can infer accurately the genetic diversity of individuals, even if you have sequencing depths as low as 1x, right? So which, if you think about it, is quite remarkable. You have, on average, one single copy per site, but you can still tell how many sites are heterozygous in a region just by integrating over all uncertainty. And in the end, if we use all these tools, we also arrive at some unbiased genotype calling. So we don't bias our genotypes towards the reference nor towards modern samples. And only this allows us to properly kind of compare ancient samples with modern samples. If we apply these tools to our samples from Aegean and Iranian first farmers, we first found that we can actually trace back the ancestry of the European first farmers directly to the Aegean, meaning that people from the Aegean spread into Europe bringing farming. However, this kind of direct line, genetic line, breaks if you go from the Aegean towards the Fertile Crescent, somewhere in between. It doesn't extend, we don't know really where, but suggesting that there was at least some cultural diffusion involved in the spread of farming. Okay, with that, I would just like to thank a lot of people involved in this work. So mostly first people from my group, Athanasios Kouzatenas, Vivian Link, a postdoc and PhD, working a lot on this. And my long-term collaborator, Chris, here is a mathematician I collaborate with a lot. Then the leading people from the paleogenetics group in Mainz who generated all the data. So this group is led by Joachim Burger. Susanna prepared all the samples in the lab for the Aegean project, okay, and also was heavily involved in many of the population genetic analysis. Farnas took the lead on the Iranian sample. Also 2 years of lab work to actually get that done, and then some analysis there. Christian is the embedded bioinformatician in the group we collaborate with a lot. And then there's a large number of people involved in these projects, archaeologists, digging out the samples, helping us with the interpretation, lots of people running analysis to make this work. Okay, and then thank you all for your attention. I'm happy to take any questions if you have some. Thanks.